Deoxyribonucleic acid is double-stranded helix which is made up of nucleotides and are linear in structure. Each nucleotide contains sugar, phosphate, and nitrogen bases. There are four types of nitrogen bases, and they are adenine, cytosine, guanine, and thymine. Adenine pairs with thymine and guanine with cytosine. DNA is found in both eukaryotic and prokaryotic cells and contain genetic information which is inherited from generation to generation. It exists in condensed and chromatin form to fit into the nucleus. Talking about histones and nucleosomes, DNA loops twice around a histone octamer to form nucleosome like beads on a string. Here, H1 histone binds to the nucleosome and to linker DNA, thereby stabilizing the chromatin fiber. Phosphate groups give DNA a negative charge. Lysine and arginine give histone a positive charge. During S phase of cell cycle, DNA and histone synthesis occur, and it is during mitosis that the chromosomes contract and become distinguishable on light microscope. Mitochondria have their own DNA, which is circular, unlike other, which is linear in structure, and does not utilize histones. Eukaryotic DNA replication is more complex than prokaryotic process. Histone acetylation is the process of removing histone's positive charge, which will cause DNA coiling to relax and increase transcription. Hence, histone acetylation makes DNA active. Histone methylation makes DNA mute. It usually causes reversible transcriptional suppression, but when there's histone deacetylation, there's removal of acetyl group from histone, which tightens the DNA coiling and decreases transcription. Chromosomes have a linear appearance and two arms. The short arm is designated as P and long arm is Q. In early part of the cell cycle, each chromosome is present as a single structure, a chromatid. During the cell cycle, the chromosome replicate and two sister chromatids form, and each chromatid contains exactly the same genetic material. There are 46 chromosomes come in 23 matching pairs and constitute the genome. One each pair come from maternal and paternal side. Out of 23 pairs, 22 are autosomes and one pair is sex chromosome. The chromatids are made up of substance called chromatin, which consists long strand of DNA wrapped around the protein called histones. Euchromatin is the part of chromosome which is less condensed, appears lighter in microscope, and are transcriptionally active, whereas heterochromatin is also a part of chromosome which is more condensed, appear dark on microscope, and are transcriptionally inactive. Centromere is the part of chromosome which is between one-third and two-thirds of the way down the chromosome. During mitosis and meiosis, the movement of chromosome on the spindle is regulated by the centromere. Kinetochore is a protein structure which assembles at the surface of the centromere, aligns chromosome, acts as a site of spindle microtubule binding, and is essential to correctly segregate sister chromatids. Telomerase is an enzyme which is made up of protein and RNA subunits that elongates chromosome by adding guanine with repetitive sequences to the end of existing chromosomes, which avoid loss of genetic material with every duplication. Hence, it protects chromosome ends from fusion and from being recognized as site of DNA damage. It is found in eukaryotic cells only and are often dysregulated in cancer cells, allowing unlimited replication. Upregulation or reactivation of telomerase is a critical feature in over 90% of the cancers and has been prime target for the development of therapeutic against cancer, as it is expressed in majority of cancer types as well as in cancer stem cells. When a few telomeres become critically shortened, this growth arrest state, and at that time, a DNA damage signal and cellular senescence is normally triggered. In the absence of other changes, cells can remain in senescent state for years, which can be considered a tumor suppressor mechanism, at least for a long-lived species such as human. It is now recognized that senescent cells can secrete factors that can influence age-associated diseases and remain viable but not dividing for long periods of time. Thus, with increased age, it is believed that there is gradual accumulation of senescent cells that may affect some aspect of aging. Telomerase also perform other important functions such as regulation of gene expression through transcriptional silencing of genes. Further studies should be performed on this enzyme for target-based therapies. As we know, nucleus is organelle within a cell which contains chromosomes, and nucleolus is the most prominent substructure of nucleus. The nucleolar organizer regions are chromosomal landmarks that consist of repeated sequence of ribosomal genes, rRNA. They are important for formation of the nucleus. In humans, the nucleolar organizer regions are typically found in the short arm of acrocentric chromosomes 13, 14, 15, 21, and 22. Here's a karyotype image of complete set of real human chromosomes. Human nucleus has 23 pairs of chromosomes. Out of 23 pairs, 22 are autosomes and one pair is sex chromosome, which is XX in female and XY in male. Autosomes are present in same number in both males and females. Structural defect of chromosomes such as deletion, duplication, and inversions are easily seen in karyotype. The metacentric chromosome is chromosome whose centromere is centrally located, which means the length of P and Q arm are almost equal. In humans, chromosome 1, 3, 16, 19, and 20 are metacentric chromosomes. Submetacentric chromosomes have slightly unequal chromosome length, for example, chromosome 2, 4, 12, 17, 18, and X chromosome. An acrocentric chromosome is chromosome where centromere is located almost close to the end of chromosome. For example, chromosome 13, 14, 15, 21, and 22.